Mutated. Question 2. 3 different mutated bacteria, X, Y, and Z, were studied. Each had a mutation in a different region of its DNA that is transcribed to rRNA. Protein synthesis was measured in cultures of each mutated bacteria, and in a culture of unmutated bacteria, results are shown. Okay, so we've got, for unmutated, you get 100% of the protein synthesis. You're mutated, don't do fabulously well. Certainly X has a really hard time if it only gets 9% of its protein synthesis occurring. Y is 15%, and Z not doing quite so badly, but still 90, so it's still going to drop. Okay, calculate the simplest whole number ratio percentage protein synthesis in cultures X, Y, and Z. Okay, not too hideous. Okay, X, Y, and Z, so in that order, 9 to 15 to 90. Okay, and then you just want to simplest whole number ratio, cancel it down, they all divide by 3, so 3 to 5 to 30. That's it. Okay. Name the other component of ribosomes apart from rRNA. Ribosomes are made of a complex made of proteins and rRNA. That's just a definition. Using your knowledge of biology, suggest how a change in the sequence of bases in DNA transcribed to rRNA in mutated cultures resulted in a decrease in protein synthesis. I think they've almost given you the answer to this, and I think that throws people when they first look at this question, because they've asked you, name the other component of ribosomes apart from rRNA. And it's proteins to make your ribosomes. But that means you need the rRNA in the ribosomes as well. So suggest how a change in the sequence in the rRNA resulted in a decrease in protein synthesis means that you could either just have your ribosomes don't form properly, and if they don't form properly, if you've not got ribosomes, and so you get less protein synthesis, that's your answer. You could also say that if the rRNA isn't there, you can't bind your mRNA properly, or that you couldn't bring in your tRNAs properly, and either of those, because they're absolutely necessary for protein synthesis, for translation, would block protein synthesis. So you just need to give a reasonable thing that could happen. Okay, describe one structural difference between DNA and rRNA. Okay, structural difference. So I'm expecting is you got 2 strands DNA, 1 strand and your rRNA. Okay, you've got your deoxyribose sugar in DNA, ribose sugar in rRNA. Okay, you're not going to get away with just saying sugar or strands. Okay, you have to give the DNA and the rRNA, and your nucleotide is your other one. Okay, so we're going thymine in DNA, uracil in rRNA. That's it. Okay, that's a question.